Thanks everyone who's joined us at this point. I expect throughout the morning we'll get a few more people joining us. This is the fourth and the last in our September webinar series. If you missed last week, we heard from Dr. Kristen Bianchini, who talked about loons and lake health. And then we also heard from James Litley, who spoke about Eurasian water milfoil management in the Okanagan Basin. If you missed those presentations and want to check them out, they are posted on the ALMS website, where you can find all the details for the webinar. And you can find all the other videos from the previous webinars there as well. This series is replacing our usual two-day in-person conference. Some of you have probably attended those conferences before. So we're just making the best of a bad situation right now. We're inviting speakers from across North America and even some excellent speakers from Alberta to share on a variety of topics. So I hope that the topics have been of interest to everyone who's joined us. Today, we're hearing from Dr. Rebecca North on winter phytoplankton, winter lakes, and from Dr. Brian Smerdon on groundwater and lake interactions. So this webinar today will be from 10 till noon. Just for logistics, the participants, you're unable to turn on your microphones or your cameras today, and the chat function has been disabled. So if you do, however, have questions, we really encourage you to ask as many questions as you'd like. You can type those into the Q&A function below. And after the presentation, Janine and Caleb and I will help to facilitate those questions. This webinar will be recorded and it will be posted on our website probably early next week. I'll just introduce as well the other panelists today. We have Janine Higgins with Alberta Environment and Parks, who's been supporting the whole webinar series with the Zoom side of things. And we have Caleb Sin from the Alberta Lake Management Society, who's going to help facilitate today. So with all the logistics out of the way, I'm excited to welcome Dr. Rebecca North from the University of Missouri, who's going to teach us a little bit about winter lakes. So I'll pass it over to you.
Thank you very much. Good morning, everybody. So today I'm going to talk to you about winter phytoplankton, dormant no more. And if your training in limnology was anything like mine, where I took my PhD degree at the University of Waterloo in Ontario, my professor Ralph Smith defined limnology as the study of lakes located within 60 miles of a major university campus, but not during the winter. And so while this is a bit tongue-in-cheek, it does kind of set the stage for the fact that we really don't know a lot about what happens to lakes in winter. And so along those lines, if we don't know, does it matter? And so what is the sound of winter, right? So if we have changes occurring during this season that is typically unmonitored and unnoticed, are we going to have any baseline data to detect any type of change? And so some literature has suggested that changes in winter related to climate change may be related to increases in summertime harmful algal blooms, or CyanoHABs, as I'll be referring to them as. It might also be related to areas of hypoxia or anoxia, so these dead zones that occur in the bottom of lakes. And of course, both of these things have implications for the whole food web and maybe declines in fisheries production. And so it's important to kind of get this baseline so we can detect any kind of change. So one of the fundamental questions being addressed right now in the literature and in research labs is, does winter set the stage for summer? And are some of these changes we're seeing in the summer, most typically related to harmful algal blooms, related to changes that are happening in the winter when we just have very little information? So this might be a familiar image to you. This is a harmful algal bloom in the western basin of Lake Erie. It is not the only situation in which we're seeing more algal blooms occurring more often in more places and for more periods of time. And so we know that there's lots of socio and economic consequences resulting from these blooms. So for example, Toledo, Ohio didn't have a safe source of drinking water for about 500,000 people, and it cost millions of dollars. And so one of the suggestions is that this increase or this change in algal blooms is something maybe climate driven, since it's happening on this global scale. And we also learned in our undergraduate limnology that there is a seasonal cycle or a traditional paradigm to phytoplankton in lakes. And so for most of this talk, I'll be referring to phytoplankton in terms of the pigment they produce, which is chlorophyll A concentrations. So this is a proxy for phytoplankton biomass. And typically we expect to see kind of two peaks, this bimodal pattern over an entire year. So one in the spring and one in the fall. And the highest biomass is occurring, in these kind of summer months. And so as a result, we design our sampling programs to capture these peaks. So we only really have a lot of information for this period of time. And the assumption is there's very low biomass and very little is happening in those winter or shoulder seasons. So now that we have this summer-centric lens, the question is, how are we going to be able to anticipate or detect any changes attributed to climate, which has the most impact in the winter months? We are starting to see more algal biomass happening in the winter. So this is an algal bloom under the ice. These are becoming more common and happening more frequently in more places. And we know that there is changes occurring to lakes in the winter. Winters are getting shorter. It is the unmonitored season, so we only really have ice data to be able to dictate how much shorter they're getting. But predictions indicate that by the end of the current century, the percentage of ice-free winters will increase from anywhere to 2 to 60%. So we are going to see lots of changes happening in the winter, and the problem is we don't have a lot of data baseline to be able to detect those changes. In a global synthesis of winter and summer data from lakes all over the world, Hampton and al. Published this figure comparing chlorophyll A concentrations in the winter to those in summer. And as you can see, the under ice chlorophyll A was about half that of summer. But if you recall my previous slide showing our assumptions, the assumption is that's much lower. So it's closer to 0 than half. And so I think an interesting take home from this is it's not 0. Chlorophyll is present in the winter and it's about half of what it is in the summer. So when we look at this from a year-round scale, if we don't account for this winter biomass, then we're underestimating chlorophyll on a year-round basis. So the objective of my talk today is just to question what happens in lakes in the winter, and then related to summer patterns, does it matter? Or are these changes we're seeing not going to have any impact on how we use and recreate on our lakes? And so, as I said, we don't have a lot of information about what's happening in the winter in lakes. The common assumption is they're dormant, so nothing is happening. It's too cold and dark for any kind of phytoplankton activity or primary productivity. And then the other, reality of it is it's very difficult logistically to sample in the winter. So we can kind of, acknowledge that something's happening, but we don't have a lot of data to support it. But we do know in certain situations, if we have the right combination of light and nutrients, we can get these under ice algal blooms. And so I'd like to try to open up this black box of the winter and shoulder seasons and determine what is the year-round phenology, so these patterns of phytoplankton biomass as chlorophyll A concentrations, some of these cyanobacteria algal toxins that they produce, because that has huge human impacts, and metabolism, so the ratio between primary productivity and respiration in lakes. And then look at this across a latitudinal gradient of lakes and reservoirs. So systems that experience winter and have ice cover, like up there in Alberta, whereas here in Missouri, ice cover is very rare, and most of our lakes don't have ice in the winter. And so if we're pessimists about this, we could make predictions that our winter futures are going to look much different based on latitude. So we call lakes up in the north that have ice cover on them dimictic. So this means they have this ice cover in the winter. As we move down latitude, as I mentioned here in Missouri, we're in the transition zone where we would call them more monomictic and we rarely get ice cover. And then we can get into situations that Europe is currently experiencing where lakes actually become permanently stratified. And so this means that this density gradient between the surface waters and the bottom waters becomes permanent year-round. And so with this, in the northern dimictic lakes, you know, I know winter seems long up there. I recently moved from Saskatchewan, but you know, we can kind of think of it as all 4 seasons are present in these types of ice-covered dimictic lakes. Whereas down in these more southern latitudes, we're getting kind of longer summers, and as with climate, we're going to get shorter winters, and then we might end up in a situation of kind of endless summers. And so this is reflected in this mean light here. So this is the light that phytoplankton experience over a 24-hour basis. In the winter, when we have ice cover, we get these convective mixing happening under the ice. And so this keeps the phytoplankton suspended up here right under the ice. And it means that over a 24-hour period, they get a decent amount of light, depending on that depth of the snow and ice, which cuts out the light from the sun into the lake. In situations where we don't have any ice cover, we don't get that inverse stratification, which means that the water column is the same density from top to bottom, which we call isothermal, and it's completely mixing. And so if you have a really deep water body, that's a deep depth or a lot of time that these phytoplankton are going to spend in these dark bottom layers away from the light. And so if there is ice cover now, we'd expect there to be, you know, a decent amount of mean light. But in these more southern latitudes, and perhaps in the future, we're going to see less light available. And then we flip again to the scenario of winters being permanently stratified, meaning that again, they're stuck in these top layers and they have a decent amount of light. And so I'm trying to simulate this kind of space per time gradient with a variety of reservoirs across the latitudinal gradient. And so these are the study systems. So for those of you that are familiar, these ones are located in Saskatchewan. The largest is Lake Diefenbaker that some people might be familiar with. We know that the ice duration on these water bodies are anywhere from 100 to over 200 ice days a year. I'm going to contrast those with Lake Simcoe down here in Ontario that I worked on as a postdoctoral fellow at Trent University, which has ice cover. It's variable, but about 70 to 100 days of ice per year. And then down here in Missouri, which I refer to as the middle of the middle of the middle, in the Midwest, we have a diversity of reservoirs that have very little ice cover. Only really the ones in the northern part of the state get any type of ice cover in the winter months. And so using this kind of gradient, we can do this space for time substitution and look at patterns in phytoplankton across these winter systems. So typically when we're looking at what controls phytoplankton or algae, we look at these 4 factors. So they are primary producers, photosynthesizers, they require light. Temperature, water temperature is important. One of the most seminal papers on cyanobacteria harmful algal blooms is called Blooms Like It Hot. Nutrients are important. So when we talk about eutrophication, we're talking about increased both nitrogen and phosphorus to our aquatic ecosystems, and that can spurge and increase algal growth. And then from kind of a top-down perspective, we have grazing. And so here I'm showing zebra mussels, but this could easily be any other type of grazer, including zooplankton. So any change in these 4 things could cause changes in algal biomass or growth. So moving from right to left, we'll start looking at grazing in the winter. We did some work on Lake Simcoe. So again, this is this large lake located just north of the city of Toronto in Ontario. Thanks to the Ontario government, there is a long-term year-round database. They took it from a drinking water treatment plant. So this is an easy, great way to work on lakes in the winter. It's basically a pipe into the lake that you can just turn on the tap and get year-round samples. And so they took actual phytoplankton samples and counted them for biomass from 1982 to 2008. And we can look at these patterns on a year-round basis. And so what we saw is that prior to the zebra mussel invasion, which was about 1995 in Lake Simcoe, we saw this kind of, bimodal pattern that we would expect out of phytoplankton. But since they invaded, you can see that there's very little pattern left to these dark solid symbols. And in fact, if anything, the maximum concentrations of phytoplankton are happening in these winter months, so January and February, which is very much ice covered on Lake Simcoe. And so one of our theories behind this paper was that this is the impact of grazing. And so Dreissenids tend to graze at faster rates with warmer water temperatures. And so during the winter, we have lower top-down pressure, and we have changes occurring in the phytoplankton biomass. The next factor that we can kind of look at controls on algal growth in the winter are nutrients. So again, looking at both nitrogen and phosphorus. I've done a lot of work looking at nutrient status indicators. So in addition to looking at the actual concentrations, we can use a variety of indicators that kind of assess the physiology of the phytoplankton to indicate phosphorus status. So deficient means they don't have enough phosphorus, sufficient means they have enough, and nitrogen. And so you can see here that under ice in all of these systems in Saskatchewan and Ontario, there was sufficient quantities of both nitrogen and phosphorus under ice available for phytoplankton. So it doesn't seem like that's something that's necessarily changing a lot in the winter or something that would be limiting their growth. So of course, the next step and what our textbooks all tell us is that there's no light in winter lakes that are ice covered. And so light is the main factor impacting their growth. And so how much light gets through into the water to access the phytoplankton will depend on whether or not there's snow presence and the depth of both the snow and ice. The type of ice also matters. And so if you're an ice fisherman or you've spent time skating on lakes, you might notice that sometimes it's really clear and you can actually see fish swimming below you. We refer to that as black ice, and it's very transparent. So the light is easily gets through that layer. And sometimes you have kind of freeze-thaw cycles and more slushy ice, which we refer to as white ice. And so that ice doesn't transmit light as much. So all of these factors will play into how much light gets into the lake. But what we also know in the winter is that with that ice cover, there's no exchange with the atmosphere. And so any oxygen that was in the lake at the beginning of the winter is all there is going to ever be at the end of the winter. And so it's just kind of a steady decline over the winter. What happens when we have changes in oxygen concentrations, and this happens most often in the summer, especially in kind of dead zones, is we have phytoplankton that grow in these surface areas. They fall down to the bottom of the lake, and through the process of respiration by bacteria that don't need light, they consume the oxygen. So we get low oxygen happening, and we can often get fish kills associated with this in the winter. What happens at that point is we get changes in redox at the sediment water interface, and things that used to be really sticky to each other, like phosphorus and iron, become released. And so this can increase the phosphorus concentration to the lake, so that legacy phosphorus or historical load of phosphorus that was safely in the bottom can come back up and be a source of phosphorus for phytoplankton. And so we know that this process happens in the summer. We call it internal phosphorus loading. And we did some work on Lake Diefenbaker in Saskatchewan to see whether or not it was happening in the winter as well. And so when we measured these input of phosphorus in the ice-covered season and the ice-free season, we found these rates of phosphorus input per meter squared per day. And when we take the ratio of them, we find that there's actually more phosphorus being loaded into the water column in the winter, 3 times more in fact, than we saw during the summer or the ice-free times. And so we know that there's sufficient amounts of nutrients in the wintertime, and perhaps this is an additional process that loads those nutrients into the lake. So now we're on to light and temperature and seeing what controls they have on algal growth. So in terms of light, we know that snow, as I mentioned, has this impact of attenuating the light. And so when the light can't get through very well, you have kind of these dark conditions or less light under the ice. When more and more light gets in as a result of less snow or changes in that black to white ice ratio, we can get these convective mixing layers showing up, which means that the algae are able to stay high in the water column and have access to light. And so we took some profiles of chlorophyll A on Lake Simcoe and saw evidence of these convective mixed layers. And so these dotted lines are the calculated convective mixed layer. So this is that density gradient. And you can see that this is where the peak in chlorophyll A is occurring. So we think that having ice is actually good in terms of light limitation. It keeps the phytoplankton up here where they can get sufficient amount of light. And then when we start losing that ice cover and you get mixing occurring throughout the water column, you're going to get less phytoplankton growth. We found when we compared snow depth, so this is the snow on top of the ice, with our readings of photosynthetically active radiation, or PAR, under the ice, that there was this significant relationship. And so obviously with more snow, we have less light. But we found there was a critical snow depth of about 3.5 centimeters. And if you have over that, then the algae are experiencing light limitations. So this dotted line is a threshold for light limitation. And you can see with that snow cover on, there's a lot of phytoplankton, or a lot of samples occurring under that point. Now we went and manually removed the snow with a shovel and measured the light again. And that's what's shown by these squares, or crosses. And you can see that under that scenario, we see a significant improvement in under ice light. So in fact, 61% increase when the snow is removed. And now most of those samples, with the exception of 5%, are above this light limitation line. And so we know that snow and changes to snow as a result of climate change will have big impacts on under ice phytoplankton and productivity. When we compare the light between lakes like dimictic reservoirs that have ice cover with ones around here in Missouri that do not get ice cover, we see some large differences. And so here I'm showing mean light. So this is the light that phytoplankton are experiencing and takes into account that mixed layer, shown as the yellow circles, and then the chlorophyll A concentrations as the green squares. And so we would expect that light would be highest and chlorophyll would be highest in the summer. Those would be our assumptions going into this. And as you can see in our dimictic reservoir, we do have chlorophyll A peaks in summer. So the maximum chlorophyll is happening during these summer months. But in this warm monomictic reservoir, there is no significant difference between chlorophyll A between seasons, so not lower in winter. And in fact, if you had to pick a maximum concentration, it actually is occurring in winter. And so this kind of goes against our assumptions of that bimodal pattern of chlorophyll A biomass. Looking at light, you can see that the highest light in the ice-covered lake is actually in the winter. But it's very variable, and this was related to how much snow. So later in the winter, we tend to lose our snow on top of the ice and it gets more windswept, and that explains this kind of variability in light. Whereas the one without ice cover, the highest light was seen in the summer. And so, in fact, when we compare these two types of reservoirs, only the summer mean light was significantly different. So there was no difference between the types for winter light. And this dimictic reservoir had twice as much light in the summer as did the warm monomictic reservoir, as a result of kind of self-shading due to phytoplankton, we assume. So when I look at these lakes on this latitudinal gradient, starting off in the north at some of these Saskatchewan Canadian reservoirs, and we look at chlorophyll A concentrations over the year, we again do not see that typical bimodal pattern that we would expect. This again is the typical sampling season that most, well, university labs even and government researchers would be conducting. And you can see that by limiting research and collection of data to the summer months, we're actually missing some of these maximum chlorophyll A concentrations, which in this case were occurring under ice right up through till the middle of March. So that's up in the north in these dimictic reservoirs. Moving a little farther south into Lake Simcoe, we see a little bit more of what we would expect in terms of the bimodal pattern. But again, these maximum concentrations of chlorophyll are happening under ice and outside of this kind of traditional sampling window. Moving farther south now here into Missouri, we see very little kind of differences on a year-round basis. But again, maximum concentrations are happening outside of our sampling window. And so I think the assumption that chlorophyll is dormant or negligible in the winter months needs to be further examined. Some daily work was done here in Columbia, Missouri, where they went out and collected samples every day, year round. Again, you see the highest peaks happening outside of that summer sampling window. So there's lots of variability, but it doesn't suggest that we should not continue to ignore winter. When I compare all these systems based on the ice cover. So here we have ice cover of less than 100 days up here, and ice cover of greater than 100 days are the two figures down here. And we separate them by winter, spring, summer, and fall. And we normalize them so that the chlorophyll A is kind of independent of nutrient concentrations and other things. We can see that in these ones where ice cover is less than 100 days, so around here in Missouri, there was significant seasonal differences, and the maximum chlorophyll is actually happening in the winter. And it's 2 times higher than what we see in the summer. Whereas in the Saskatchewan and sometimes on Lake Simcoe, we can see there's less differences between these seasonalities. So ice cover does play a strong role in what we expect to see in terms of chlorophyll in the winter. So I think we need to, question these traditional paradigms and start thinking about opening up our sampling season to incorporate more of the winter. And I think this becomes critically important when we start addressing how green is too green, and why are we concerned? Why do we care if we're missing these peaks in chlorophyll? And so unfortunately, sometimes with the chlorophyll, that can be dominated by cyanobacteria, which are kind of the bad type of phytoplankton and have the ability to produce toxins. So these are just some news articles where we're seeing more and more cases of toxic water or algal toxins impacting drinking water, impacting aquatic organisms, and potentially getting into the food web. And so one of the ways we identify whether or not water is safe to swim in or drink out of is its color, right? So we assume that the greener it is means the more chlorophyll or phytoplankton is present, and it could potentially be toxic. And these are the types of warnings or signs that are often put up to have people avoid interacting with these waters that have the ability to produce toxins. And obviously there's some serious negative impacts to having these toxins being produced. We can get exposed to them in many different routes, in fact. So even if you don't directly swim in the water, you can still inhale them. They can aerosolize during recreational activities. You can also get them through the food web. And so there's been work showing that they actually go through the food web, and you can consume them in contaminated fish. And also, of course, what we've heard about most often with Toledo and other places is this consumption in drinking water. Most of the time, our drinking water processes are really effective to remove the toxins, but as we saw in Toledo, sometimes it just overwhelms the system and the demand. So one of the things I had to deal with as being a new assistant professor is how does one sample a variety of lakes on a year-round basis? And I think so far, the answer appears to be community scientists. And so we initiated a high school program called Reservoir Observer Student Scientists, or ROS, where we have high school students as part of their class and their curriculum go out to a local lake or reservoir, ideally close to where their school is, and collect a weekly water sample. They take it back to their labs and classrooms and filter it and preserve it and freeze it. And then we do all the analysis in my lab at the University of Missouri. So this has been recently funded by the North Central Region Water Network and has allowed us to expand it up to a latitudinal gradient. So from Missouri up to Minnesota and Wisconsin, though today I'll just be talking about some of the data from Missouri. And so this is work by my master's student, Emily Kinzinger. And so she's looking at this year-round chlorophyll A concentrations, the algal toxin microcystin, and the algal toxin selenospirmopsin, and comparing concentrations in the non-summer and the summer seasons. And so again, it is rare to get ice cover around here. So these reservoirs are mostly this warm monomictic. And so when we go into this, our assumption would have been that chlorophyll and toxins would have been significantly higher in the summer. That is how we monitor, and that is how we regulate our water bodies. And so her results so far are pretty surprising. You can see here for chlorophyll A concentrations in all 3 of these reservoirs, there was no significant difference in the chlorophyll between the non-summer and the summer months. And so the letters above these box plots indicate significance. If they're the same, they're not significantly different. And if they're different letters like an A and a B, then that's a significant difference. So no difference between summer and winter in terms of chlorophyll. In terms of the toxin microcystin, again, we see no significant difference for 2 out of the 3 from summer to winter. But on this one reservoir, which is a local urban reservoir here in Columbia, we see significantly higher microcystin happening in the non-summer months. And so again, this is typically when people are not concerned. Stevens, for example, is not a drinking water reservoir, but it does have a recreational beach on it, whereas McDaniel is a drinking water reservoir. So, you know, understanding these differences and what's happening in the winter can change how we mitigate harmful algal blooms occurring and monitor for these toxins. The other toxin, spermopsin, is not as common. And so you can see we barely detected it in 2 out of our 3 reservoirs. But in this urban pond that was sampled by high school students, we can see that there's significantly higher toxins happening in the non-summer months. So in all cases for both toxins, the highest concentrations we observed of both toxins were in the non-summer season. Here's some detailed data from this Bethel Lake. Now I'm showing microcystin and spermopsin in the blue and green circles over multiple years, year round. And you can see that they have opposite relationships. So it tends to be when cylindro is high, microcystin's low, and vice versa. With spermopsin, we're seeing the highest concentrations we ever recorded in the non-summer season. So this is October, November, December. And microcystin, we're seeing the maximum concentrations happening in the summer. And so there's this kind of opposite relationship to these 2 toxins and suggestions that we shouldn't dismiss these off-season CyanoHAB events. I should mention that these concentrations are significantly below the EPA's recommended limits. So here in the US for microcystin, it's 8 micrograms per liter, and spermopsin is 15 micrograms per liter. So you can see my scales are nowhere near that. So these are well below these kind of recreational thresholds, but still, I think, interesting that the highest concentrations are happening in these off seasons. And so this lack of relationship between both chlorophyll A and toxins and between 2 different types of toxins suggests that the green might not be a good indicator of toxic waters. This is very much yin and yang relationship between microcystin and spermopsin. If you're monitoring for microcystin or for cyanotoxins, you're most often monitoring for microcystin. It's the most common and ubiquitous. But we have suggestions that spermopsin concentrations, while low overall, are peaking in the clear waters and in the winter seasons relative to microcystin. And so this suggests that we can't make assumptions about positive relationships between any of these factors. The next question people want to know about phytoplankton in the winter is, are they productive? So we talked so far about biomass and the production of cyanotoxins. Metabolism or primary productivity is also really important in terms of both greenhouse gas emissions and carbon flux, and also food webs. And so here I'm showing under ice data with chlorophyll A concentrations on the x-axis and aerial gross or primary productivity, so how much oxygen is being produced by the phytoplankton, on the y-axis. And you can see in this case, green does seem to be productive. So there's a significant positive relationship between these 2. It's not very strong, but there does seem to be that positive relationship. When we compare production rates with respiration, so aerial respiration is the consumption of oxygen, production is the production of oxygen, and we look at them in these under ice systems from days since ice on, we can see these relationships. So productivity is shown in the black circles and respiration is showing by the X's again here. So we can see that days since ice on, the productivity, you know, kind of shows a bit of a pattern with peaks happening kind of at the end of the winter. And that's what I mentioned tends to be the time when the snow blows off and we get more black ice. And so that may be related to that. And we can see that while respiration is fairly low, it does kind of show some peaks happening at the beginning of these winter months. When we compare them for a year-round basis, so, you know, does this matter? Does changes in winter have impacts on summer? When we look at the data under ice by these X's down here versus open water, you can see that it doesn't even compare. So most of the time, you know, the open water is following this 1-to-1 line between production and respiration, whereas winter tends to have really low production relative to summer. If values fall above this line, so the P to R ratio is above 1, we consider this to be autotrophic. And below that line, we consider it to be heterotrophic. And so again, the common assumption with low light in the winter is that most lakes in the winter are heterotrophic, right? So they're not producing any new biomass. And so this follows true. The open water rates were 17 to 66 times greater than the under ice rates. So, you know, while there was a positive relationship between productivity and chlorophyll, it doesn't appear that that winter chlorophyll is that productive or is producing that much oxygen. In addition, the respiration, which often follows water temperatures, tends to be 8 times higher in the open water season relative to the under ice. So while it's interesting that there is production happening at all under ice, relative to what we see in terms of summer rates, it's very minimal. And so to kind of summarize what happens in lakes in the winter, or what we've learned so far, there appears to be sufficient quantity and quality of nutrients. And so we know that there's mechanisms that are, you know, making maybe nutrients more available. There's also less demand for those nutrients in the winter months. And so it seems like, you know, any changes to nutrients may not have that big of an impact on the phytoplankton that live there. And this refers to both nitrogen and phosphorus. And one process that may be contributing to that is this wintertime internal nutrient loading. We do know that there's low under ice light due to snow cover. I didn't show the data, but I do have some physiological photosynthetic parameters that indicate that while they are light limited, if light conditions improve, they will respond. So they have the ability or the energy, you can think of it that way, to respond to increases in light. So again, with climate change, if we get less snow cover occurring, we could see a quick response in terms of phytoplankton to that increased light in terms of increased productivity and biomass. So we do see these under ice peaks in phytoplankton. So chlorophyll A wasn't significantly lower in the winter, but it didn't seem to be that productive relative to summer. And so maybe that, you know, what sound does winter make or how is it connected to the summer, may be more related to that biomass that's produced in the winter as opposed to its primary productivity. Though it was interesting to note that on a few occasions, typically towards the end of the winter when that snow blew off, they did switch to autotrophic conditions. So production was greater than respiration under those circumstances. So it can occur under ice, and maybe as we get to a future of less snow, we'll see that happening more often. So there does appear to be some evidence of shifting peaks in terms of changes occurring to the winter, which indicates that there is some consequence to phytoplankton and summertime conditions as related to these climate-induced shorter winters. Again, if you want to be a pessimist, we are maybe starting to transition to kind of these warm monomictic conditions. I have colleagues and I've read research of people working on, you know, what happens in both terms and culturally and recreationally and financially when we don't have ice cover anymore in our northern lakes. And as I mentioned, in Europe, they've, you know, Lake Zurich, for example, has already gone beyond that. So it hasn't had ice cover in a decade. And now it's starting to look like it's permanently stratifying. So it's already going through that transition that I suggested at the beginning. There is, I think, as these things shift, the potential for more and different cyanotoxins. And so again, we're most often looking for microcystin, but if we're seeing more selenospramopsin or other toxins occurring in the winter, and we're not monitoring for them, we could totally miss that. And again, with that lack of baseline data, how will we know that there's been a change? And so I think understanding and having this baseline data is pretty critical, and now is the time if we want to try to make any kind of prediction about what's going to happen under our future winter scenario. And this is going to have implications for year-round lake function, not just winter. So does winter matter? I would suggest yes. Unfortunately, we're getting shorter winters, and so we need to understand now what implications these changes will have. And so currently, I think we are missing pieces of the puzzle, notably in the fall and spring or the shoulder seasons and in the winter. And this assumption that winters are dormant maybe should be tossed out the window, so to speak. So we're seeing rapidly changing light and physical mixing dynamics in lakes, and these can be linked and facilitate cyanobacterial growth and then potentially the production of cyanotoxins. And so we should be more vigilant about these changes, especially in drinking water sources. So with that, I would like to acknowledge the various sources of funding for the research that has, well, been decades long that I covered today. And thank you for your attention, and open it up to any questions that anyone might have put in the chat.
Awesome. Thanks so much for the presentation. And I'll just add on to the end of that, that the Alberta Lake Management Society, for those who aren't aware, we do have a winter monitoring program that's been going for just 2 years now, where we've been partnering with anglers and stewards out at lakes throughout the winter to collect samples from about December to March. So if you're interested in that program and want to help collect more data and help us better understand our winter lakes, keep an eye on our newsletter. We'll be announcing in the coming months how you can get your hands on one of our sampling kits. So we do have 2 questions that were sent in. One question, which I believe you touched on, but it just, and maybe if you want to add anything else to it, it's regarding the importance of internal phosphorus loading in the wintertime for a year-round nutrient budget. Do you have anything else you wanted to add to that?
Yeah, I think we don't know in larger systems. So the data I showed was for Lake Diefenbaker in Saskatchewan. And to my knowledge, there's only been like maybe that work done on 5 water bodies in all of Canada. We did a review paper a couple of years ago, Diane Oriel led, where we kind of compared these things and found very little data. But the data that we did find suggests that yes, it can occur, and especially in smaller systems that have that permanent ice cover. So, you know, fish kills are not uncommon. And so, and in fact, you know, in most of the northern provinces, they don't even stock a lot of lakes because they know that there's going to be this fish kill by the end. So those would be the systems that would be, I would look at first in terms of the importance of internal phosphorus loading. But I think in Lake Diefenbaker, what we found is, yeah, it can really impact, it can be higher, in fact, than what we saw in the summer. And so from a year-round basis, kind of balancing that out could be important.
Great, thanks. And then there's another question here. I'm just going to try and maybe reword it a little bit, but do you have a sense of if the winter phytoplankton assemblages and biomass are indicators of what your summer is going to look like?
Yeah, so in a couple of ways, they can be kind of cedar populations. And so, you know, when we go into summer, that reserve of phytoplankton could go in and decide who wins first in the competition of phytoplankton within the lake. And we can also see changes, you know, to the oxygen. So again, if we have this large production of biomass happening in the winter, and then that sinks, and then in the summer starts undergoing decomposition, we could see, you know, kind of more occasions where we have dead zones or low oxygen in those bottom waters. And so the more biomass in the winter, the more there is to fuel those types of dead zones. And that would obviously have implications.
Do you have a sense of which species you're seeing, especially when you talk about cylindrospermopsin? Do you see some patterns with what species are producing?
Yeah, I think that is like the biggest question right now in this. So no, the reason we use chlorophyll is because it's a quick and dirty kind of proxy for phytoplankton biomass. Cost, you know, a couple dollars a sample. To get a trained phycologist to identify samples under a microscope can run you close to $250 a sample. So for these large kind of monitoring programs, you know, I personally don't have that kind of species level data. And what I have seen on other lakes and in the literature is all over the place. So, you know, I know the story on Lake Baikal is related to diatoms, and you know, it's the diatoms that are coming up under the ice and having these large blooms, and it's happening in different seasonal patterns, different years and things. Other cases, it's green algae. Like basically every time I read a paper where they talk about what type of algae is in the winter, it's a different type. So I feel like that's something that is wide open, and it would be really great to see some more work done on that.
Great, thanks. And I see we have a question that's come in. It says, you've referred to climate change several times. What do you use as an indicator? Here in central Alberta, there's virtually no evidence of change during the satellite data era.
So a lot of the ice cover information is either, historical records. I think one of the best examples I've seen, and Sapna Sharma has done a lot of this work at the University of Toronto, is there used to be like cultural festivals that happened on the ice. And so they could go back through the records and figure out what years and when that festival was happening based on how thick the ice was. And so there's documented historical cultural changes like that we have evidence of. So any kind of, record where people have kept track of ice shows a decline. And then we do have, now we're using satellite imagery and other things to kind of show that as well. So I don't know the specifics about Alberta. I've never worked in Alberta, but I know on a global basis that is the trend.
And I will say there is a program out of Ontario, I believe it's called Ice Watch. And if anyone watching likes to record ice on, ice off data, you can upload that information to IceWatch, or you can contact us and we'll help you to get that data uploaded.
Yeah, another plug on that, there's also a citizen science app through GLEON, the Global Lake Environmental Observatory Network. And so it's to, a same kind of thing. And we've just added, or 2 years ago, they added ice on, ice off information and thickness. So another place that as community citizens, you guys can go in and record that information, and it can go up and be used on global patterns by lots of different scientists.
Great, thanks. And we have another question here. Is the toxin cylindrocermopsin specific to a single species, or can multiple species produce this toxin?
Yeah, that's the tricky part about cyanobacteria. So in all cases, multiple types of phytoplankton can produce multiple types of toxins. And so it is impossible to link the presence of a toxin to an individual species. So often, for microcystin, we think of microcystis, which is a phytoplankton species, as producing that, but we know that there's a variety of different phytoplankton out there that can produce it. So it's really hard to link that. One of the culprits that we often think about in terms of cylindrospermopsin is Cylindrospermopsis, which is a type of phytoplankton, and it used to be thought of as a tropical phytoplankton. And so, it didn't used to be here in Missouri in the Midwest, and now it's pretty prominent, actually. And I know in Canada, it's varied whether or not it can be found in samples and located. So maybe that's, kind of an invasive species or global change thing happening as well with our phytoplankton species, which might be why we're seeing more of these toxins in more places.
Great, thank you.
Are there any questions from Caleb or Janine? I know Caleb always has a good question.
Yeah, I've got a question. Have you looked at the spatial variability of chlorophyll on winter lakes, like across the lake as a surface? Like, have you seen differences in chlorophyll for that kind of sampling?
Yeah, no, I haven't looked. So anyone who's done winter sampling can appreciate how difficult it is, and to be able to get to all those spots, in the same day, month, whatever, to make those kind of maps. Lake Simcoe, we did, I think, 13 stations on the lake, but it's a 600 square kilometer lake. So it's, they're all just spot points. So I haven't seen any good work. And the good work that's coming out of that kind of spatial resolution is typically done now with like satellite imagery. And again, with that ice cover, we can't get at that information. And so, yeah, I think that would be interesting to know. And that would go along with these physical mixing dynamics as well, to know what those flow patterns are like under the ice. But I'm not aware of any good spatial coverage on a lake in winter for mostly logistical reasons.
Great. Maybe to kind of follow up on that, when you're taking these chlorophyll samples, are you collecting them like right beneath the ice, or is there kind of like an optimum like below the ice where you're trying to take the sample, or where are the like samples vertically taken?
Yeah, so from a research perspective, we cheat in that we have a YSI that measures chlorophyll, like that one profile I showed. And so typically what we did is we profiled first, watched it, kind of live, and then sampled for the peak. But typically that peak is happening in that layer, not directly under the ice, but like say a meter below the ice. So if you had to pick a depth, I would say, kind of go a meter below that ice surface. Now it's complicated, right? Because as soon as you auger that hole, then all the water is going to flush up through the hole and kind of mix. So like on Lake Diefenbaker, the ice was 1 meter thick. So you're kind of disrupting those layers and that pattern as soon as you put a hole in and try to sample. So I don't think we can really be that specific. We kind of just went for, you know, close to the ice, underneath the ice.
All right, and for the Ross program, are the students just taking a sample from the edge of the lake, or are they actually taking a boat onto the lake and sampling from a specific location?
They're taking it from the edge of the lake. Yeah, we didn't have the the paperwork involved in putting high school students in boats. Yeah, we are not doing it that way. We're doing it if there's a dock. In some cases, on some lakes, there is a dock that goes quite out into the lake, and so that's perfect and easy to sample off of. And then one of the reasons that I only showed Missouri data is because, again, we don't have ice cover here. So we have the most data from these southern locations because the students, you know, even if there's a thin layer, they can usually take a stick or something and get through and get a water sample. Our sample this last winter was our first year in Minnesota. And there's less samples there because it was more work to go in and auger the holes and do that. And in that case, it was the teacher who actually went out and did the on-ice work, and then the students did the filtering back in the lab. So yes, winter is complicated and difficult, and these are shoreline samples where we just give them a sampler to collect water, surface water. So just, you know, we tell them to avoid kind of scums or anything directly that you can see on the surface, but not to dig into the mud either. So we're calling it the epilimnium, but it's definitely a shoreline sample.
Thanks.
Great, thanks. In Alberta, we've had a couple cases where Planktothrix blooms under the ice and gets trapped even as the ice is forming. Have you come across that in your own work?
Definitely, yeah. So Planktothrix, for the rest of you, is red. And I've heard great stories of, like, you know, there's a body under the ice because it looks like blood in some situations. And it does grow up through those cracks and breaks in the ice. And so, yes, I've seen Planktothrix along that latitudinal gradient. So we have pictures here in Missouri. Here under the ice, it looks like kind of flowers, like it kind of, you know, blooms out, maybe like snowflake kind of patterns. And then I know certainly up in Toronto and in Ontario area, there was lots of that happening as well. And it does produce toxins. So this is another scenario where we have these toxin producing ones happening in the winter under ice.
Great. Any other questions from anyone on the panel or anyone viewing today? You can type those into the Q&A. Okay, I don't think so. So I just want to say thank you for the great presentation. It's a lot of food for thought for our own winter monitoring program coming up this season here in Alberta. And it was a fantastic presentation. So thank you so much for taking the time to do that for us.
Thank you for having me. Great.
So I think what we'll do is move on to our next presentation. I believe our speaker is here. So for those who've just joined us, the chat function is disabled, but as we go through our next presentation, you can type your questions into the Q&A function. And at the end of the presentation, we will help facilitate those to our next speaker. So our next speaker is Dr. Brian Smerdon from the Alberta Geological Survey, and we've asked him to come in today and talk a little bit about the groundwater-lake interaction. As you can see from the title, it's a hidden part of the lake ecosystem and not always something that's at the top of mind. So I'm excited to welcome Dr. Brian Smerdon, and I think I'll just pass it over to you.
Perfect. Well, thanks for the introduction, and yeah, thanks for the invitation to join the group today and talk a little bit about this. Yeah, I guess getting right into it is hidden part of the lake ecosystem, and perhaps just to, oops, hold on a sec. There we go. Jump right in. It's often considered, I guess, a hidden part of the water cycle in itself. So just as a reminder, this image on the screen now sort of shows a classic kind of water cycle that you may have seen in, high school, university, or wherever. Really, it's depicting kind of what you'd see across Alberta. So you got, mountains on the west, plains heading towards the east, This particular diagram really showing like a lot of the surface hydrology that we're pretty familiar with, lakes and rivers, those things you can see pretty well, of course, you know, evaporation happening, but underlying it all, this whole thing about groundwater that occurs on many scales, which plays a role in terms of lakes and rivers, and we'll get into that a little bit today. In getting the slides together for this webinar, I was sort of trolling back through the literature. It's been a while since I looked at some of the lake work, and was really pleased to see a pair of review papers. So mostly led by Don Rosenberry out of the USGS. And so I just thought I'd put them here on this slide to kind of set the stage. His words are pretty similar to the title of this talk, of course, a disregarded component of the lake and water budget. So maybe a little bit stronger terminology, but I encourage you, if you're interested, to learn more about some of the things you might hear today, or just to learn more about groundwater in general and its relationship to sort of lake hydrology. These are worth checking out. Feel free to contact me if you're having trouble getting them, but they do sort of set the stage. And of course, they're done in parallel, one being on the hydrology and one being on the nutrients. Okay, so these are basically borrowed straight from that first paper, but they resonated with me, so I thought they'd be a nice way to introduce what we're sort of getting into. They kind of set up this review with reasons why you would neglect groundwater, or reasons why it's been neglected over the years. And there was a list of about 7 or 8 points, but I sort of grouped a few of them here, which you can kind of see in this tan colored box. So I guess the first is it's invisible. And that kind of goes without saying. There's a lot of societies and groups that work with groundwater around the world right now, really pushing to make the invisible visible. That's actually a slogan for some of them, is to really try to shine the spotlight on it where it's an important component of the water cycle, where it has you know, implications for various pieces of work. Second is, I guess, with regards to inflows and outflows. They're often considered very small. So if you're doing a lake budget, or if you're just looking at the hydrology of an area, if the budgets, you know, shows that these inflows and outflows are relatively small, maybe you can consider that a zero balance, and it's almost not worth considering. I certainly have, you know, hydrological engineers as colleagues that claim you can get many lakes and rivers in Alberta to balance without even considering groundwater at all, which is an interesting comment, but we'll see it actually plays a fairly significant role when delivering things like phosphorus to a lake. Third point, hard to measure directly, and that goes back to the invisibility comment. It's very difficult. You know, if you grew up on a farm, you have a well, or you've seen springs, certainly you can see evidence of it, but it's not something that comes to top of mind when you think about hydrology. It's often considered quite complex. Water tables move up and down with the season. You know, you get drought or overpumping that influences things, and so it's quite variable, which again, feeding back to that invisibility factor, it's just hard to kind of conceptualize. And then there were a few other bullet points that I grouped together into this thing, decided to call discipline compartmentalization. And that's both from a research perspective or a training perspective, but also in terms of management. So to be completely honest, my bias comes from the geosciences. I grew up in an earth science setting, discovered groundwater that way, but then very quickly realized the water cycle's one thing, so we need to consider all of it. Often people come at their career or their work from one area or another, and you may not get a chance to see what's happening in other parts of it. Interestingly, though, that carries over to management as well. A lot of policies are set for surface water, or they're set for groundwater. They may not be for both. There are jurisdictions in the world that do try to account and manage these as a single resource, which is an admirable task. Now, where we'll hopefully get the thoughts to in this webinar is that importance is, it creates a really unique ecotone. So at the surface and groundwater interface, right where everything is kind of happening, you get lots of activity that's important for lake bed ecosystem. You get important sort of chemical reactions happening, oxidation, redox conditions, nutrient transfers, like all sorts of really neat things happening at a very small scale right where this interface occurs. And that's actually quite a delicate interface. So as things change, as climate changes, as pumping can happen, as the lake itself is used for other purposes, some of those changes can really disrupt that interface. So it becomes quite important helped to at least understand how some of these things work together. Now, I've already fully declared my, I guess, bias and upbringing. I am a groundwater scientist, so I'll start with this piece of it. And when I, earlier in my career, had been working with a forest hydrologist, we landed on this discussion of, well, isn't groundwater just water below the ground? And to some degree, that's true. It's a very easy way to visualize it. But thinking about the formal definition actually helps a little bit to kind of understand that it goes beyond that. So the kind of classic definition that has emerged in the last sort of 50 years is that, really it's in this zone of saturation. And I guess that's important. And if you can kind of see on the left-hand side of this image, there's a really zoomed in version where you may have sediments, down below on the bottom part, these sediments are totally saturated with water. And so that would be where groundwater lives. It's completely full of water. Above that, you get to the limit where there's the water table. That's the top part of this saturation zone. And then above that, you get into pores that are not quite filled with water, or they're filled with air. And that becomes important because it differentiates a lot of biogeochemical reactions that occur, but then it really sets up the sort of flow system that I'll speak about in a few minutes. So if we look to the image on the right, it's kind of a classic textbook diagram where you would have an elevated area that we call a recharge area. So precipitation entering into the ground surface, percolating through that unsaturated zone and becoming part of the saturated zone, becoming groundwater. It can then move. So it moves no differently than if you were to pour water on the side of a hill slope from high to low areas. Could move quickly, could move relatively quickly through the subsurface towards a discharge area. So in this case, it's shown as just a creek or a river. That may happen in a few days, may happen in a few years, relatively short times in the hydrology sense of things. The unique feature about groundwater is that it could also then take a very much longer flow path. So it could migrate its way based on the geology sort of down through an aquifer system, crossing these lower permeability layers, traveling for really long periods of time. So in some cases, centuries or millennia, like this can be quite a long period of time before it eventually comes out again in that discharge area. This starts to paint a picture of what the challenge is in thinking about groundwater, not just that it's invisible, but that has some really long time frames attached with it as well. So to kind of go way back, and this is a couple of slides borrowed from sort of university lecture material, 1940s. So this is a person by the name of M. King Hubbert, who worked in the United States, really set kind of the stage for how groundwater would move. So I just spoke about these recharge and discharge areas. And what he did was actually take a look at some of the mathematics behind it, which I'm not going to go into, but really it borrows a lot from how mass is transferred, or how heat is transferred, or in some cases how electricity is transferred. They're all sort of governed by the same kind of equation, which again I'm not going to show. But what sparked for him, I think, was to show this in terms of a field, or an energy potential field. Now this is kind of like a fictitious cross-section. You can imagine it as being sort of like a landscape between two rivers. And his depiction of it, with the math behind it as well, would show that you have this hydraulic potential field in the subsurface. So if you go to the top of the hill and you were to drill, let's say 3 wells side by side, or all very close together, you would find that as the wells are deeper, the water levels tend to be deeper in those wells as well. And what that tells us is that, from a sort of potential theory, is that water wants to move down. So that's the recharge zone. Water's coming in, it's really wanting to move down into the subsurface and kind of get away from the water table and get away from the land surface. And that sets up the condition for the first part of the flow system. What that looks like in the real world, of course, these are just a couple of images from the sort of north of Edson area. It's just material that's permeable. So in the upper image, it's just a gravel capped upland. Water likes to move through that, moves through that pretty readily. On the bottom part, what's shown is actually an outcrop of the Pascapoo Formation. So that's a geological formation that spans from kind of Hinton, arcs its way down through kind of Drayton Valley, all the way to Calgary. But it's a lovely sandstone in much of its, in some parts of its body. And that transmits water very readily as well. So those are just examples of what they could look like, out in the real world. On the flip side, sort of down in the valley, if you were to kind of do the same thing, drill 3 wells again, we find that you really get the opposite condition. You have these deeper wells that actually have a very high water level. And if you have a shallower well, the water level is even lower. So in some cases, these can be what's known as artesian. You may have heard that word before, or you have water kind of flowing out the top of the well, or the water level is above the ground surface. And that's really telling you that the water in this location in the subsurface wants to move upwards. It's trying to get back out. So in this case, it's heading towards this feature that's a river or a lake. Now, when M. King Hubbert did this work, it was quite theoretical. He did actually grow up in a farming community, so part of the work was to look for water and where to go looking for it. The theory sort of ignited, I think, a lot of folks that were living in the prairie settings, and Alberta is no exception, to use this idea to start to look for where water could be, and it actually worked out quite well. And the other pioneer behind this work, coming on about 20 years later, was Joseph Toth, who was an emeritus professor at the U of A. Okay, so discharge features. Now, they tend to be a little more interesting looking. You're probably familiar with springs. And so on the top image, that's Big Hill Springs, kind of down near Calgary. This is basically formed because of the geology that's there and the fact that it's an area where groundwater is coming to the surface and it's gushing out. It's quite spectacular. The other feature is the Elk Island silt holes. So that's shown on the image down below. I think I took that picture a couple years ago on one of our tremendously smoky days, when it's got that hazy appearance. Same process is happening here, except it's not gushing out of the ground. It's basically just trickling. It's coming up very, very slowly, sort of feeding these soft, squishy silt hole features. If you've seen them in Elk Island, you could think of them effectively, it's quicksand. And so in the middle part of the image, there's a tiny little bit of water. There's some reeds growing around it, and that's a discharge area. Now, throughout the, I guess, 1970s and 1980s, mostly across the prairies, but driven largely by some work at the USGS, folks were taking these ideas and thinking about, well, how would that work with lakes? And so there's a very classic piece of literature from the USGS called, I think it's Groundwater and Surface Water, a Single Resource. It's 1998. Thomas Winter was the lead author on that. And one of the images, so the one I've shown here on the left-hand side, is just how would that translate into kind of the understanding of lakes. So the top image A is gaining lake. You've got high water table all around it. Groundwater is feeding into the lake, so it's discharging into the lake. The opposite condition, the middle panel, is a losing lake. So you've got basically lakes that are kind of mounded up and they're sort of pushing water back out into the landscape where you have groundwater recharge occurring. And then, of course, you can get combinations of these things where what's known as a flow-through lake. So you could have this thinking that water coming in one part of the lake area and then leaving at a different part of the lake surface. And that's kind of a combination, much more interesting to think about, and a lot of things happening, quite dynamic with that. So while the USGS was putting a lot of this work together, and many researchers in other parts of the world, I think the limnology side of things was also tapping into this idea. And so Catherine Webster published a few papers early on this idea about landscape position. And so what's shown here is an image that basically combines some of these ideas around recharge and discharge and how it could look in a landscape setting. So if you have elevated areas, so shown sort of up here, high landscape position, the lakes are tending to lose water. Down in the low area, low landscape position, they're gaining water. And then of course in the middle, you see these combinations where you have groundwater interacting with different lakes along the way. What I think made sense for that thread of research was really to start explaining some observations that were coming in terms of, well, how do the lakes function in terms of their chemical budget? How do they respond to drought? How do they respond to change? And this idea or this notion about having the different kinds of lakes in a landscape position that's shown here started to explain that. So a lot of the work kind of in the late 80s, early 90s around drought would show, of course, these high landscape, mounded lakes were very susceptible to drought. When they lost a lot of their surface water, there wasn't any groundwater to help supply them. So that would then have implications for the ecosystem within and surrounding that lake. Whereas lakes lower in the landscape position, of course, quite resilient. They're effectively buffered from a lot of this change, and they're able to kind of maintain themselves over much longer periods when there could be drought. And the other thing this shows is that, of course, this is all a connected water resource. And so it's critical to think about it in that way, because the ecosystem kind of runs hand in hand with how the water is moving through the landscape. Now, the two review papers do an excellent job sort of setting the stage and the background. One of the points that Don Rosenberry makes in the first review paper is that, and you can see that some of this literature is from kind of the mid-90s, is that the interest in it was quick. It grew very quickly, but what they sort of recommend, or what they, I guess, conclude, was that the actual tools and some of the analysis techniques to measure some of this interaction was a little bit delayed. There were some early things that worked really well, but it didn't grow as fast as the interest did. So a classic case where I guess the science is trying to catch up with the interest, which keeps things interesting, I guess. So I thought for the next part of this webinar, we could take a look at just some of the techniques that are used to really look at lake budgets and how groundwater can interact with the lake. So what's shown here first is what's known as the residual approach. Pretty conceptually straightforward. So if we look at this sort of depiction of the lake being a perfectly nice bowl, it's really just to measure the inflows and outflows that you can see, or that you can get a hold of pretty readily. Things like precipitation, shown in blue here, any surface water coming into it, evaporation leaving, and of course surface water going out. Those are relatively straightforward to measure. So the picture on the bottom is just an image from a lake that I had studied quite some time ago. We've got a rain gauge in the middle, a couple of evaporation pans, and we were measuring these kinds of things. If you measure the lake stage and you know something about the bathymetry, you can estimate the volume. And so then you have a change in volume as well, as you're looking at it month to month or year to year, whatever the case may be. So adding up the amount coming in, knowing something about the amount going out, and knowing something about how the volume or the change in storage may have evolved, you'll get left over with a bit of a residual, things that don't quite line up. And often in this residual approach, you just attribute that to say, well, it probably has something to do with groundwater. Now, as I mentioned earlier, a lot of very simple models will end up with a residual close to zero. And that would lead you to think, you can measure everything really well. That's great. Residual is very low. Maybe it doesn't interact with groundwater. This feature doesn't interact with groundwater very much. Or it could be higher, and that gets you thinking, well, something else is going on. The other thing to keep in mind about this is that, this is actually a fairly simple rain gauge shown in this picture, but there's many ways to measure precipitation, many ways to estimate evaporation. Even simple act of trying to measure creeks discharging into a river can be complicated. So all the little errors that come into those estimates and summations eventually get translated to the residual in this case. So it leads you to perhaps think the groundwater is more or less important depending on how those errors shake out. Now, of course, this can be improved with not just tracking the volume of water, but trying to track things like the mass of phosphorus coming into and out of a lake, other nutrients. It can do it with stable isotopes, which I'll talk about in a few minutes' time. That's kind of the starting point. The second step, or a little bit more advanced, is called the Darcy approach. And so up on the right-hand side, if we go back to thinking about, yes, the lake's sitting within a groundwater flow field of some sort, maybe we can measure that somehow. And so if we have our same lake here, kind of shown with the bucket again, off on the left-hand side, this is just a schematic to say, well, what if there were a couple of wells drilled nearby the lake, and we could estimate from the water level what the slope of the water table would be kind of heading into the lake. And let's say it's sloping into the lake. So it's kind of like option A up here. So if we know the slope from the couple of wells or more, we know what the substrate looks like. Is it permeable or is it not permeable? You can do a fairly simple calculation using Darcy's law, which is an old, the fundamental of equations in groundwater science, but fairly simple, to come up with how much volume of water is coming into the lake. So sort of that discharge flux coming into it. It works fairly well. You can break up the shoreline into segments. You could do it on, multiple segments coming into the lake, leading out of the lake. It helps generate some understanding of what those fluxes could be, which in theory helps refine the budget, helps understand if a connection that's happening there, helps get away from what the residual would be. Of course, all these would not sum up to zero, but it's a starting point. But it's that key piece in the subsurface then. What's it made of? Because some material will transmit water very readily, and some will not transmit water as readily. So it's really dependent on the permeability of that material. Now, what's shown here on the bottom right-hand side is just a snapshot of, it's a schematic really, but the permeability of the subsurface varies by orders of magnitude, depending on where you're in. And when you say that to people, they start to think, what? Orders of magnitude. So when we talk about something like this Pascapoo formation that I mentioned earlier. So these are just units of how permeable, so the hydraulic conductivity of that material could be. And these are orders of magnitude changes. So many of these and most porous media will span easily 2 or 3 orders of magnitude. So to kind of visualize how different that is, you're talking centimeter to meter to kilometer, right? That's 3 orders of magnitude. And we're using that kind of number in here to estimate the flux coming in and out of the lake. So obviously we're introducing another bit of uncertainty, but it's really doing kind of the best job you can. Now, as an example, this is an older example, from my doctoral work quite some time ago now. We did this for a series of lakes up in the Uticuma area, so near Red Earth Creek, just north of Uticuma Lake itself. It's just a series of small lakes. The one that I sort of focused on was number 16, had a romantic name of Lake 16. It's about 1 kilometer across or so. This is a glacial sort of outwash landscape. It's quite coarse. We put in, I don't know, probably close to 100 wells, mostly by hand, around the lake to try some of these techniques of getting the budget for this lake. So what's shown in the upper panel is contours of the water table. You can kind of see it's wrapping itself around the lake. There's water feeding into it from the south, from the north, from the east. This series of lakes exists kind of in that landscape position. So if we look at this red cross section that's shown on the upper image, the cross section is shown on the right, or sorry, shown below. The lake kind of in the middle. It's a little bit higher on the east side, a little bit lower on the west side. It sets up that classic kind of flow through style lake. Now all these interpretations, I guess, or inferences about water movement were made from the wells we had put in. So we were able to break up the shoreline, do the water budget, and start to learn about this particular lake. That's fine in a research setting where you can send out grad students and put in wells and that kind of thing. The other way we've started to do this is just utilizing parts of the province that have water wells or enough water wells around them. So this is actually, apologies, it's probably a little hard to see because it's smaller, but Cardinal Lake, so just near Grimshaw, near Priest River. This is a Grimshaw aquifer system that's there. It's a shallow gravel deposit which interacts with this lake and supplies a lot of folks with water. So what's happened here is you have wells that are drilled into the formation over the course of decades to supply water, and every time a well gets drilled, the driller submits the record to the province. The record tells you what the water level was when they completed the well. And so what we can do is take that and start to map out what the water table would look like around the lake and start to think about, well, how does this lake interact with this shallow groundwater deposit? So what's shown here is we've just broken it up into decades. And so we've got 60s, 70s, 80s, 90s, kind of through to today. Really, in this case, the wells are not sufficiently close to the lake to get the kind of resolution you would get from a research type approach, but it does show us that water's moving from these White Mud Hills down towards the Peace Valley. As it gets close to the lake or nearby it, it does interact with it, but the lake itself is then supplying water back into the groundwater as well. It's kind of an interesting way to sort of catalog that. Now, the other way to look at interactions, I guess, with a lake between groundwater is to try to look at the seepage directly. So as I said earlier, it's groundwater's not only invisible, but difficult to measure directly. So in the mid to late 70s, a lot of folks started to look at how to do this. And the really low-tech way was with a seepage meter. So I guess the advantage is that with the way lakes are configured often, the shoreline tends to have the highest amount of seepage when it intersects with where the groundwater is feeding into it. Tends to be lower as you get into the deeper portions of the lake. It's not always true, but it just means you can wander out in waders and actually do some work. So what's shown here kind of in this schematic would just be lake surface, lake bed down below, and then a seepage meter. Now it's a really sort of glorified term for, you know, the top, or so the bottom part of a bucket that's been cut off, or a barrel, or anything that you could kind of shove into the subsurface, and then have a little stopper with a bag attached to it. Very high-tech Home Depot approach to measuring seepage. But what happens, and it works pretty well, is that if you have seepage coming into the lake, basically it fills up the bag, so you can know the area of the circumference of the area of your device, the volume of the bag, and the time, and then you actually can start to estimate how much flux is coming into it, so discharge. It theoretically works if water is leaving the lake as well. You can fill up the bag and watch it get pulled out. Tends not to work quite as well, but it does theoretically work. What's shown on the right, just again, you know, in theory, this is quite good to do. It's used quite a lot across the prairies. In practice, you do have to find spots in the lake bed where you can put these kinds of meters in. And so the little disc-shaped thing is the bottom of a food-grade barrel that's been cut to do this. Of course, you've got riparian, you know, vegetation happening all around here as well. So it you know, can be challenging, but it does work. A very low-tech way to look at seepage. Now, kind of moving through the literature and through time, the other way to look at seepage, the most promising, has been actually with using a lot of thermal methods. Now, this could have been done previously just simply with thermistors and you know, simple temperature sensors. That's the history of it. But what's really evolved probably in the last kind of 10 years is a lot of the handheld devices. So like these FLIR, almost camera-like imagery, you know, sort of sensors where, you know, folks are pointing them at the wall to figure out of your home where you might need more insulation. You can use it to map a lot of really interesting things in nature this way. And so in terms of groundwater discharge, I've seen some folks that have actually used it to look at springs, or where you have seepage coming into rivers and lakes. That's pretty cool. I don't have an image to show that here, but I'm sure you can get the idea for it. What I wanted to show was a bit of work that's using these DTS systems. So this is actually a fiber optic cable, tremendously long, that's laid out, you know, in various settings to look at temperature or distributed temperature across that array. So this example is just borrowed from a paper. The work's done actually in Denmark. So you can look at the sort of panel B here. It's a small lake with its surface catchment. You can see they've actually mapped out, this is from well data, what the water table would look like feeding into the lake. And then they kind of zoom in on one of the pieces of the lakeshore to show an application of this temperature sensor. And so what's shown in panel D, I guess would be, is this really long squiggly yellow line. And so these are often hundreds of meters long, fiber optic cable, basically, which gets laid out on the lake bed and then used to measure temperature. So down in panel A, I think this was a sort of early summer temperature snapshot. You can kind of see the warmer colors being a bit warmer, hotter temperatures, the bluer colors being cooler temperatures. And so at the same time, they also have some seepage estimates in the same location. And what's shown on this panel on the right-hand side is where the seepage patterns were calculated from the temperature and also from some seepage estimate. So it's a really nice way to look at in quite high resolution where you've actually got groundwater feeding up into the lake. Now, what the imagery shows beautifully is that it's of course not uniform. It's quite variable spatially, you know, but it is coming in. It does have a flux, and it probably has a fairly important role in this case in terms of the lake's chemistry. The temperature aspect is interesting because that then makes a nice link to habitat characterization. Where do you need certain thermal refuge to occur? You know, this type of approach lets you start to look at that as well, which is pretty cool. Not something I've ever worked with. Now, another approach would be to work in kind of model space, in that if you have an understanding in a three-dimensional sense of what the lake looks like, the geology that it's sitting in, and you can start to assemble some of this in a flow model. And so what's shown here is just some work some colleagues at the Alberta Geological Survey have done. So starting kind of up here at Sylvan Lake is this little lake shown in the middle, and the catchment around it. So the Sylvan Lake subbasin, basically, Medicine and Blind Man rivers. And what they did was build or construct a three-dimensional representation of the geology, and then applied sort of hydraulic boundary conditions like rainfall coming in, and simulate what the flow field would look like. And so what's shown in the colors, the sort of red colors are where the water table is high, blue is lower, flow is from high to low. We take a slice through the middle of it, right through Sylvan Lake. You can barely see in the middle of this cross-section where Sylvan Lake would be, but it's kind of bounded between these tiny little uplands, and the simulation shows that you get water moving from the uplands down towards the lake from both sides of it, but that you also get some water moving down deeper into the flow field and heading off kind of towards the east. Some of it discharging in the Red Deer River, some of it discharging farther off into the plains. So it's really showing then that lake as part of its position in its local landscape, but then also the context of kind of the bigger sort of hydrogeological representation. On the left-hand side, it's just a zoom in to sort of look at that and how this could be useful in terms of understanding lake and groundwater connectivity. And so again, we've got the outline of Sylvan Lake shown here. The high colors showing where the water table would be higher, blue colors being lower. And really what this helps you do then is define, well, what would the capture zone be from a subsurface perspective? So where is the lake able to capture groundwater from? How far away? And what would that look like? Which is quite a good, I guess, result of doing this kind of a modeling exercise, in that, you know, yes, the model's calibrated against real world observation, but it lets you start to explore, you know, well, what would this look like? And then that feeds into discussion about, well, how's landscape being used? Does this kind of tool fit into that discussion as well? And in this case, we see with Sylvan Lake that a portion of its shoreline actually is recharging water back into the groundwater and flowing off towards towards the northeast. So quite an interesting way to put pieces together. Now, this might be familiar, depending how much of this type of thing you've seen before, because quite early on in Alberta's history with looking at lakes and groundwater, I think this was in Ellie Previs's group quite a while ago at U of A. They did some work on Narrow Lake, and I thought this was an interesting one to share. The paper's from 1990, thesis a little bit before that, but it was one of the early examples, even almost globally, where you had a combination of different methods coming together to try to look at a problem. So they were able to actually go in and drill some holes, do the research thing, use multiple methods to look at seepage, residual approach, the Darcy approach, because they could calculate it, seepage meters, and because of someone was at U of A at the same time, I think it was Frank Schwartz, had some early computer simulation, which is shown on the right-hand side here. You know, classic 1980s groundwater flow simulation to show how would water come into this lake. Now, from a lot of this work, the conclusions were the budget of the lake itself, you know, really less than a third of it was coming from groundwater, but from parallel study of the phosphorus setting, it was really the dominant source of phosphorus feeding into the sort of upper portions of the lake, which was quite cool. And that goes back to a comment I made earlier that, you know, just because you're working with a residual budget that might tell you that the components of the water budget are very small, they can be quite critical when it comes to the lake chemistry. Okay, so to shift gears a little bit, the next part of this will just look at some of the other methods that have been used. And I want to revisit the sort of lake budget again, but this time looking at it through the lens of the stable isotopes. So isotopes of water itself. Now, this is expressed in this kind of funny terminology, if you've never seen it before, called delta 18O, or delta hydrogen, delta deuterium. And really what this is a ratio between oxygen 18 and 16, and then deuterium, so 2 hydrogen atoms and 1. And that's the water molecule itself. Now, in hydrology, this is nothing new. This has been around for decades, because the ratio of those two different oxygen isotopes and those two different hydrogen isotopes changes due to temperature, so changes because of evaporation. So it's very useful when you're trying to track things. Now, really the key player in terms of Alberta, of course, has been John Gibson, and he's done a phenomenal amount of work in this space to look at applying this, using these isotopes to look at the lake budget. Now, what's shown here is a little bit more hairy. I kind of like the simple diagram, but it's the same idea. If you can measure the stable isotope ratios of all these same components, you can add that to your water budget and get a better understanding of the lake. John sort of took this kind of a step further early in his research, and it's been applied across lots of Alberta, actually. His model sort of, you can see the same kind of thing. You're really trying to track the isotopic signature of water coming in, water leaving, and its interaction with the atmosphere, because it does evaporate. So there's a few things that have to be considered. But the way he sort of draws the model together and has refined it over the years is that if you can collect a water sample from the lake, almost on its own now, it can really, through the modeling that he's developed over the decades, is to really define the ratio of evaporation to inflow. So as lakes evaporate, they tend to get themselves enriched in the heavier isotopes, and they lose the lighter isotopes to the atmosphere. And so as you sample a bunch of lakes, you start to get a better feel for what that evaporation ratio could be for the amount of groundwater coming in, or water coming in for that matter. And then his model goes on to then estimate, well, how would that get to that stage in terms of the water yield? And then you can even start to look at, well, what kind of residence time would this lake have? And I think they go on to define what residence time is in terms of turnover. Now, this is probably familiar to a few people in this group, because it was done for some 50 lakes, I think, across most of the North Saskatchewan basin, or at least in the Edmonton area. So the approach of using this isotopic mass balance is really useful for regional classification. You can go out, it's a lot of work to sample lakes, but you can go out, sample a bunch of lakes, get the analysis done, which is not rocket science anymore, and run it through the model. So it's really helpful then to generate some metrics around that inflow-outflow ratio, residence time, etc. And then if you think one step further, when you start to look at those differences, that might tell you where you have perhaps groundwater being important for some of these lakes. It certainly won't be important for all of them, but it will be definitely important for some of them. And you can tease that out of things like the residence time and the evaporation to inflow ratio. Okay, probably the newest sort of technique to come into the surface and groundwater literature, definitely into lakes, has been the application of radon. Now, you're thinking radon, the stuff that hopefully is not in my basement, right? That's right, it's the same thing. So radon as a gas, as a material, is generated by all geological media. It's everywhere. The challenge in the basement setting is that if the sediments where your home is built is generating a bunch of radon, and it comes into your home and you're not able to sort of get rid of it, then it accumulates and over long term can unfortunately cause cancer, apparently. In a tracer setting, it's another tool for the toolbox. So it's an interesting tool because you can take the same approach where you can start to look at a lake and say, well, do we see radon in this lake? Do we see radon in the groundwater near it? Do we see radon in the surface water? It's a bit of a different tracer though, because it does decay over time. So it takes just about a little less than 4 days to run through a half-life. So if it can accumulate, it means it's coming in at a rate that's higher than, of course, the rate that it would be decaying. So again, we can go from my sort of simple image to something a little bit more complicated. And this is some work I think that comes out of Germany. I think John Gibson was actually involved in this as well. What's shown here, again, is just a lake setting. And so we've got the kind of bowl for a lake, let's say, but this is now viewed through the generation and movement of radon. So if we start kind of at the bottom, this is actually a field site, so there were measurements, but you can measure radon in groundwater. You can measure the concentration, and maybe that's one of the sources of radon coming into the lake. So that's a number. You can measure radon in the lake itself. You can do profiles, much like you would see in a lot of other lake work, where you have radon coming in, or basically the inventory. So how much radon is sitting in that water column. Of course, it could also diffuse just from the sediments that the lake is sitting in, just on a much slower time scale, but that can happen as well. And all that radon moves up into the lake, it starts to decay, right? Every 3.8 days, there's half as much as there was before, but it's also still coming in at the same time. So that sets up the kind of inventory piece. The other thing that happens with the atmosphere, because it is a gas, is that it will then come out of solution and basically dissolve, or the dissolved gas comes out of the water as it gets to the top of the water column and heads off into the atmosphere and basically degasses and moves away. The radon tends to be quite low in the atmosphere because it gets blown around and it keeps decaying and basically disappears. So what this sets up though is another way to look at how to do a budget for a lake, because you have different sources, an inventory, and some really well-known relationships. And so people can build, again, models from that. Now, I think one of the last kind of graphics we'll show is probably, unfortunately, one of the most complicated looking ones, but that's what this other group had done. Again, I think this is in Germany as well. What's shown on the upper panel, so this is time, okay? Time across the horizontal axes. And what's shown on the upper panel is the stable isotope concentration. And so, of course, I think this is these shaded bars on the very top are precipitation. But so throughout the summer months, so this is kind of like July, August, September, October, the stable isotopes, their ratio is increasing. So whatever lake this is, it's evaporating. The stable isotope ratio is increasing. You see that around the world. That's nothing kind of new. Then they enter an ice-covered period. Doesn't interact with the atmosphere anymore. So the stable isotope signature starts to change and it decreases again. Okay, that's fine. What's shown on the bottom, this is actually now the radon concentration of the lake itself. And so you can see for the summer months, it's got a concentration that sort of goes up and down a little bit. But then what's interesting is that during that ice cover period, it starts to increase. And so with the model that was developed for this to explain both the stable isotopes of water and the radon itself, is that during that ice cover period, this lake still has groundwater feeding into it. And so that does a couple of things. It keeps delivering radon into the lake, but because the lake is ice capped, it can't degas as quickly as it did before. Now it can still decay, and it will always decay, but it tends to start to accumulate then. And so then they can then model, and which I've kind of highlighted down below in this little yellow shading, is the result of how much groundwater would have to come in, knowing all the different concentrations, to maintain this observation. And it's actually a very kind of clever way to look at how much groundwater could be feeding into this lake. Now, this might start to ring a few bells with a couple people, perhaps, in that the other way to look at lakes is that during that ice, and taking advantage of the ice cover period, which you heard a little bit about in the last talk as well, is just by simply measuring what was the lake level right before it froze and what was the lake level right after it thaws. And in these lakes that accumulate groundwater, if you can catch that measurement at just the right time, you actually see that the lake level could increase. Saw that in some of the lakes we were working at in my PhD work, and I know some colleagues have tried to do that at some of the lakes in Parkland County as well, just as a surrogate for doing a big seepage estimate. It's really taking advantage of simple observation. But this is how it could look for using radon. Okay, radon kind of brings us almost full circle back to the geology piece, because radon's generated by all geologic material. Like I said before, it's kind of everywhere, hopefully not in your home. And I mentioned earlier about the permeability. And so not all formations, of course, are the same. And in going back full circle, what's the role of geology in this lake question? And it's really then saying, well, where is the lake sitting? So it's sitting in a certain place in its landscape, but it's also sitting in some aspect of the subsurface as well. And so what's beneath of it? Now, quite simply, you can think about the geology or the hydrogeology in terms of being aquifers, so materials that tend to move water very readily, or aquitards, things that don't move water as readily. So aquifers would be things like sand and gravel, sandstone, bedrock, whereas the aquitards tend to be more of the clay-rich material that just doesn't transmit water as readily. It does, but very, very slowly. Now, the challenge, I guess, with the subsurface and anything in geosciences is shown by this photograph on the lower right-hand side. This is just actually from just close to the Drayton Valley Bridge. And that's the North Saskatchewan River right in front. And it's again a shot of the Paskapoo Formation. And what it very clearly shows is that, it's a mixed bag out there. One particular formation is going to have pieces in it that are sandy. It's going to have pieces in it that are clay. That's just the way the deposition occurred, a long time ago. And that's what we're kind of left with today. But learning something about that actually then helps understand the connection between groundwaters and lakes. And this slide here basically just borrows a bit of work from Alex Oifer, who's at AEP. Previously, he had done some work with, I think it was the Sturgeon River Basin, so it's reported on in the North Saskatchewan Watershed Alliance. But he looked at some water levels in some of the lakes in Parkland County, but then trying to relate their fluctuations to what they were sitting in. So what he mapped out here was a few of these lakes in cross-section, and ones that are better connected to sandy sediments and ones that are less connected to sandy sediments. And what he started to see was that the relationship between the fluctuating lake levels does have a role, or does have a link to the geological material of the substrate that it's sitting in. So it kind of brings us full circle to needing to know something about the landscape, needing to know something about the geology, and then understanding some of those things together will sort of paint a bigger picture for the lake and groundwater question. So to kind of wrap up this tour de force through groundwater and lakes, yes, it may be hidden, or it is hidden, but it does interact with lakes, and lots of literature starts to show that. That degree of interaction is quite variable. Sometimes it's not tremendously strong, sometimes it's very well connected. But most of the time, it's actually quite important for the function of the lake when you start to go beyond just the water budget. Its response to drought can be governed by landscape position. Its sort of chemical signature can be governed by how much phosphorus is coming in or how much of a nutrient is coming in. The other challenging aspect is that, as you may have a bit of a taste for now, those methods of investigation have grown over the years, but they also vary quite widely. So no one technique will be the sort of silver bullet to understanding how groundwater is connected with a lake, but multiple lines of evidence do. And I think the Shaw paper showed that quite early in the history of this research, and certainly many others now that are combining radon with other measurements to get a better understanding of what's happening in these lakes. And then I guess the last point is that to sort of tie it all together, and that I do come from a geological survey, is that subsurface framework does play a key role. It sort of sets the stage for what's happening. And yeah, that's kind of the introduction to groundwater and lakes. So thanks.
Great, thanks so much for the presentation. I see we have at least one question in the Q&A. If you do have questions, feel free to type them into the Q&A box there, and we'll get to them. And we've got some questions for you ourselves. So Janine and Caleb and I will help navigate through those.
Okay.
Great. So the first question that we have here is that there is a tall veg running under Gull Lake. Could it be providing water to the lake?
I didn't catch the first part of that. There is
a tall veg. Tall veg?
Okay.
Saying that right, Brad?
From what I know about Gull Lake, which is pretty limited, but yeah, I think that is the case. You could have that. I think I've seen that in some reporting as well in terms of groundwater feeding into Gull. I think some of the modeling showed that as well. It's similar to Sylvan Lake in that regard, but slightly different configuration.
Okay, great, thank you. When modeling groundwater seepage during winter, you mentioned that there is a stage increase after winter, which would indicate groundwater inputs during ice-on periods. Is this due to a discrepancy from winter precipitation and expected stage after ice off?
Very good point. Actually, excellent point and a very good question. In the presentation was probably playing a little fast and loose with some of the requirements, but yes, if you do have an understanding of the snow accumulation on top of the ice bed, on top of the lake ice itself, that does need to be considered in the calculation as well. It's I guess where I was going with it is that it's a measurement that can be made relatively simply. If you know there's a lot of winter precipitation or snow, you probably do need to consider that in terms of it being useful. But again, it's another of the single pieces of evidence that may help understand what's happening with a lake. Not in itself foolproof, because as pointed out by the question, you do need to know what's happening over the course of the winter, but a relatively straightforward thing to try to do.
Okay, we have another question here. They say, good synthesis of all the methods. Which one does AGS use the most?
We, well, I guess we tend not to look at specific lakes in general. We tend to, so if we're working at a more regional scale, so the Grimshaw example is a good one, we would tend to use sort of more the Darcy approach, because from a desktop perspective, we can get at a lot of data fairly easily. If we start to dive down into having to do nitty-gritty specific lake research, that sort of falls out of what the survey would normally do, but a lot of the isotope methods I know colleagues at the U of A have been using, they seem to work very well, especially the stable isotope methods. We've been, so I've been working with them because we do have a radon detector at the AGS. We've been working with them to try to better, I guess, generate some information around radon in the lakes as well. Hopefully that answered the question.
Great, thank you. Somebody else says, thanks, Brian. Could you outline some of these techniques could you outline how some of these techniques informed the Alberta government surface water directive that was applied to lakes?
Unfortunately, I don't know if I can do that, because I will not know all the details of that. I'm sorry to say. If there's anyone else on the panel that might know something about that, feel free to jump in.
We can always follow up with this person after the fact as well, if we can track someone down who can answer it. All right, we will go to another question here. Is there historical data of phosphorus levels in groundwater throughout Alberta?
Yes, so the two data sources that are readily available. One is that AEP have a groundwater observation well network. There's 250 some wells across the province that are sampled periodically, not every year. There are some that are sampled quite frequently and some less so, but that information would usually have a lot of parameters, including phosphorus. The second would be Alberta Health. So if homeowners have sampled a well out of their curiosity, I think sometimes you could see phosphorus reported in that work as well. Not all the time, but it does exist.
Sorry, can you just clarify the website or the portal that people could access to see that information?
Sure. You could try to take a look at, it's AEPHIN, AEPHIN. If you Google that, that will take you to the Alberta Health portal that has groundwater information on it. Off the top of my head, I can't remember if phosphorus is one of the ones that they have directly on that portal, but sometimes the underlying data could have that as well. And then the other one is the Groundwater Observation Network. So if you type GOWN, you can find that as well for Alberta. And probably a whole bunch of stores that sell clothing. But
I just put the AEPHIN website in the chat to everybody, so people should be able to access that there.
I have a question. There's kind of a belief, or you hear at lakes sometimes that people believe the groundwater is, for some reason, cleaner, I'll say, than the lake water. But you mentioned an example where groundwater was a huge source of phosphorus for a lake system. Can you just comment on that, and maybe what the chemistry of groundwater tends to be in comparison to lakes?
Good question, because that feeds back into the spatial sort of variability. There, generally speaking, groundwater quality tends to be quite good. It's harder to get at in some cases compared to surface water. Now, with regards to lakes, it then depends, I guess, how much turnover you would have and other activity in the lake. So there could be lots of lakes that have, like algal blooms and various things that are going to deter in terms of the water quality. In that case, groundwater quality might be a bit better or preferred, but groundwater quality is also variable across the province. So some parts of the province will have, in thinking as you get towards sort of the eastern part of the province, Provost area, south of Provost, those tend to have quite well, some of the water is good. I'm not going to say it's all bad water, but you tend to get a lot more salts in it and a lot more solid, totally dissolved solids, which influences the water quality. So where we see really good water quality tends to be kind of in the Pascapoo, let's say, because it's replenished very readily from being close to the mountains, get lots of rainfall and snow, and that changes as you move across the plains.
Okay, great. And I see we have another question here. Have any groundwater dependent municipalities used your work to limit population expansion?
Not that I'm aware of. I don't, yeah. I know we're working now with, so through Alberta Innovates funded project, their water innovation program, working with EPCOR and the University of Alberta on better understanding the relationship between groundwater in the Edmonton area, which we haven't looked at in a very, very long time, and the river. And so I don't think there's a population cap behind that work. It's more just making sure that there is longer term supply for a growing population.
Great, thank you.
I'm curious if the capture zone of a lake necessarily matches up with the watershed area, that the surface watershed area.
Yeah, that's a good question. Most often it doesn't, because it's the controlling factors have more to do with the shape of the basin and then the geological setting itself, which sometimes can be the same configuration as the surface catchment, but more often than not is different. So the PhD example actually even and the Sylvan Lake example both show capture zones that have not a random, but a different looking shape than you might imagine. And it's not related to the surface catchment as much.
Right, so we have another question here. In some cases, large groundwater withdrawals can change a lake from a receiving lake to a discharge lake. Is there a way to predict this on a large scale to better manage impacts of groundwater use on surface water?
Yeah, great question. That would start to tread into being able, yeah, you could do some simple modeling exercises. So they don't necessarily have to be full blown and three-dimensional, but even from working with some more simple equations, kind of an engineering approach where you could look at how much the pumping could influence let's say the water table configuration or the hydraulic head, and then whether that, depending on how far away this pumping was to a lake source, what the kind of time frame to capture could be. That would be the simple end of things. More complicated, which actually is not that complicated, but more complicated would be to build kind of a more 3D type model or look at it in a three-dimensional setting and see what sort of volumes would then start to influence the lake condition.
Caleb, do you want to ask any questions?
Yeah, I'm curious about some of the isotopes. If you could use the isotopes to measure over time how groundwater influence to lakes might change, if you're using heavy water or radon. Is that possible or something that's been done before?
Yeah, and again, most of it has been done, and I think it can be quite informative. It tends, you greatly expand your field program that way, but can learn a fair bit. I know in a lot of John Gibson's earlier work, it was more synoptic sampling. So, flying from lake to lake or getting from lake to lake quickly and just doing a whole big region. But I think he did have some earlier papers where they looked at time series. I know in my work, both in my PhD work, but then also now when we're looking at rivers, having a bit of a time series behind it helps, I think, help understand that, what could be changing through time, and then you can start to extrapolate how that impacts the lake.
Lakes are quite high this year, and I think there's probably a lot of, I'm just guessing, there's probably some groundwater recharge happening in a lot of these systems. It sounds like from your presentation that there's not a surefire way to look at a lake's water chemistry and say, oh, there's a lot of sulfate in this lake, must have a high groundwater impact. Can you just elaborate a bit? It sounds like you really need to know what your groundwater is like to understand what that's going to look like in the lake. I know, for example, in the Beaver River Basin, we have high arsenic levels in the groundwater, and we tend to see those in the lakes as well. So that seems to be a good indicator, but it's probably pretty unique to every system, hey?
Yeah, it's probably a little bit unique. I think it's the way that I think about this is that lakes and rivers are relatively easy to sample. You can get to them. You can acquire a sample pretty, without too much effort. And then it tells you something. It's then interpreting what you're seeing that becomes the challenge. So in the case of the arsenic that you mentioned, if you saw it in the lake, natural question would be, well, how much is in the groundwater, right? So then you'd have to start looking for places to sample from groundwater as well. As I mentioned earlier, the province does have observation network, but it's only 250 wells spread across the whole province, and most of them are sort of Edmonton and south. There's a lot in Fort McMurray for other reasons as well. It then becomes a challenge of finding a way to get that measurement. So it's sort of a tiered approach where, yes, you can find something in a river or lake and then want to learn more about what it means. Quite often in our work now, it's, I don't want to say it's impossible, but it's difficult to go out and run a big groundwater sampling program. Certainly, the province has done that in the past with regards to like when coal bed methane was a big thing. 10 years ago, there was lots of baseline work, or more, I should say. But it then becomes a reason to look for places to sample groundwater, right? So Grimshaw, I guess another example where we were able to work with kind of a local group, the municipal, they have an aquifer management association there that facilitated us being able to sample some groundwater at the same time as the lake. So that was fairly helpful. I'm not sure if this is getting at an answer to your question yet.
No, I think that's good. I think it does kind of answer it in the sense that you need to look at, what's in the lake and then look at what's in the groundwater and look for that connection. So,
yeah, I think the other aspect that comes into this would be the more citizen science approach too, in that if there's interest in an area and, you have some observations from lake or shallow groundwater that are starting to generate more questions, is that, quite often there's other folks that live in the same area that may be they may have a well, they may have, you may be able to start to access that. And so the sort of citizen side can come in and say, well, now we have all these other places we can acquire samples from to learn about a region which may not have much information.
Are there any other questions from anyone on the panel today? No? Okay. So thanks so much for the presentation. I think it was great to get us again thinking about groundwater because, like you said, it's out of sight, so it's often out of mind, but it is an important part of our lake system. So thanks for taking the time to do that for us today.
Great. Well, yeah, thank you for having me.
And of course, thank you to everyone who's viewed the presentations over the past 4 weeks. I have to thank all of our great speakers. We had 8 speakers who gave great presentations on a variety of topics. You can still view those all on our website, and today's presentation will be posted online as well. Of course, thanks to Alberta Environment and Parks for hosting us through Zoom. Jenna and Janine, thanks for all your help with that. And of course, thanks to Caleb for helping to facilitate a number of these presentations. With that, I think we'll say goodbye to everyone, and hopefully we'll see you at another webinar or hopefully in-person conference next year.
Okay, thanks.
Bye, everyone.
Bye.
Thanks, Brad.